All cells need energy. Energy flows into an ecosystem as sunlight and ultimately leaves as heat. ATP powers most cellular work and is produced during this process. Cellular respiration includes both aerobic and anaerobic, but is often used to refer to aerobic respiration. Although carbohydrates, fats, and proteins are all consumed as fuel, it is helpful to trace cellular respiration with the sugar glucose. Through glucose plus oxygen yields carbon dioxide, water, and energy as the form of ATP and heat. But first, a little review of chemistry. Chemical reactions that transfer electrons between reactants are called oxidation reduction reactions or redox reactions. The reactants are what you start with. The products are the ending result. When a molecule becomes oxidized, it is losing electrons. When a molecule becomes reduced, it gains electrons. Energy harvest through NADH and the electron chain are coupled to efficiently harness energy. NADH will pass the electrons to the electron transport chain as oxygen pulls electrons down the chain in an energy tumble. The energy yielded is used to regenerate ATP. In this redox reaction, NAD, shown here, receives hydrogen ions from food, as shown here. With the aid of an enzyme, dehydrogenase, these electrons and protons of H+ are transferred from the food molecules, such as glucose, into our NAD, which becomes reduced with the addition of the H+ to form NADH. Let's begin talking about cellular respiration. Cellular respiration begins with glycolysis, then pyruvate oxidation in the citric acid cycle, ending with electron transport and chemiosmosis of oxidative phosphorylation. All living things need energy, and production typically starts with glycolysis. Glycolysis happens in the cytosol and produces pyruvate and ATP at substrate level phosphorylation from glucose. Then pyruvate, our 3-carbon molecule, is oxidized to become acetyl-CoA, a 4-carbon molecule. Acetyl-CoA then enters the citric acid cycle to become electron carriers of NADH and FADH2, as well as producing a small amount of ATP. In the last step, these electron-carrying molecules from the citric acid cycle, as well as glycolysis, enter oxidative phosphorylation, which consists of the electron transport and chemiosmosis. During this step, the bulk of ATP is produced through oxidative phosphorylation. Let's begin with glycolysis. In the investment and payoff phases of glycolysis, you can see ATP is used to produce ATP as well as NADH. All cells use glycolysis even if oxygen is unavailable, because oxygen is not needed for this step. the resulting products of ATP, NADH, and pyruvate can be used for future reactions. If no oxygen is available, cells will undergo fermentation and anaerobic respiration. They can use this to produce ATP without oxygen. Most cellular respiration requires oxygen to produce ATP. Without oxygen, the electron transport chain will cease to operate. In that case, glycolysis will couple with anaerobic respiration or fermentation to produce ATP. There are several types of fermentation. All start with glycolysis producing NADH and pyruvate or pyruvic acid. There are several organisms which can undergo fermentation. No ATP is produced in this process, but they are regenerating NAD+ for glycolysis. Additional products include carbon dioxide through several fermentation methods, as well as lactic acid that we see in cheese, yogurt, and soy sauce, ethanol in alcohol products, acetone as cleaners, and acetic acid in vinegar. Fermentation is also used to produce products like Swiss cheese. Returning to aerobic respiration with oxygen, we now will pick up where we left off with our pyruvate. The pyruvate will be oxidized during pyruvate oxidation. During this step, the products of glycolysis in the cytosol are entering the mitochondria. A carbon dioxide is released. Coenzyme A is used to produce acetyl-CoA, a new 3-carbon sugar that will be used in the citric acid cycle. The acetyl-CoA will enter the mitochondrial matrix inside the inner mitochondrial membrane to begin the Krebs or citric acid cycle. The Krebs or citric acid cycle starts with oxaloacetate as it joins acetyl-CoA, thereby regenerating our acetyl-CoA enzyme. As we proceed through this cycle, NADH, ATP, and FADH are produced. During this process, carbon molecules are increased and decreased during this regenerative cycle. As the energy molecules of NADH and FADH2 are reduced, they are accepting electrons when the intermediates carbon molecules are oxidized and carbon dioxide is lost. These energy molecules of NADH and FADH2 enter oxidative phosphorylation and flow through the electron transport chain. In the electron transport chain, NADH and FADH2 travel through a series of cytochrome proteins. The cytochromes accept the hydrogen ions and shuttle them into the intermembrane space of the mitochondria. The electron transport chain is coupled with ATP synthase for chemiosmosis, where the bulk of ATP is made. Oxygen is acting as a terminal electron acceptor in aerobic respiration and will form water in combination with some of the hydrogens that were donated by NADH and FADH2. The remaining hydrogen ions create a proton motive force, or a gradient, of hydrogen plus ions. During chemiosmosis, these hydrogen ions flow through ATP synthase into the inner membrane space. As this happens, ATP harnesses this energy, this proton motive force, to drive the oxidative phosphorylation of ATP. In summary, glycolysis produces 2-pyruvate, 2-ATP, and 2-NADH. These molecules are used later on. The pyruvate becomes acetyl-CoA that enters the citric acid cycle. The citric acid cycle produces 2-ATP as well as additional NADH and FADH2. All of the NADH and FADH2 molecules from the prior steps enter oxidative phosphorylation to produce an additional 26 or 28 ATP molecules. In total, per glucose molecule, 30 to 32 molecules of ATP are produced. However, not all of we eat is glucose. What is this significant about the location of oxidative phosphorylation, and where do we get energy from other foods? Many nutrients are used for oxidative phosphorylation and ATP. Complex carbohydrates can be broken down into sugars and enter glycolysis directly. Proteins are broken down into amino acids and then enter cellular respiration at different stages, either as pyruvate, acetyl-CoA, or directly into the citric acid cycle. One waste product from this step is ammonia, NH3. Fats, on the other hand, are broken down into their components of glycerol and fatty acids. The glycerol molecules become G3P, or glyceraldehyde 3-phosphate, during the conversion of glucose to pyruvate and can enter cellular respiration. Fatty acids, on the other hand, are long carbohydrate chains that will contribute to the production of acetyl-CoA. All of these products will then enter into the citric acid cycle and then into oxidative phosphorylation. Different food products have different levels of calories or energy based upon how large those molecules are and how many carbon atoms are contributing to this energy production.